Hello everyone, welcome back to Bioinformatics BB. This is Dr. Babajan Puranganapalli, Associate Professor in Bioinformatics. First, I want to sincere apology for not uploading video for such a long time. Life has been incredibly busy, but during this break, I have learned lots about the bioinformatics and AI advancement. So these advancements has been evolutionized how we can handle the data, so making analysis more faster, more accessible, and more efficient, even for the large scale of data sets. Okay, so I am thrilled to back and excited to share everything I have learned. Together, we can explore various tools, workflows, and extract a meaningful insight into the bioinformatics. Okay, so let's dive into our new series on gene expression analysis. Today's session, we are going to learn about the basic web-based platform GEO to R, how we can perform the gene expression analysis. Okay, so let's start. So if you see the basic introduction to the gene expression analysis, so gene expression referred to as a process where the genetic information is converted into the functional molecule like the RNA or protein. Okay, so by comparing the gene expression levels between the groups such as healthy and diseased tissues, we can easily identify the differentially expressed gene that is called as DGs. So these DGs often reveals insight into disease mechanism, pathways, and they acts as a therapeutic targets for us. Okay, so many applications are there for the gene expression analysis. These are widely used in the medicine, drug discovery, and biology to understand the molecular mechanism of it. Okay, and today's we are going to do hands-on approach how we can explore, how we can study this gene expression analysis by using the various tools. So we already know that one there are so many methods are available in gene expression. You can see the quantitative PCR, microarray, RNA-seq, northern blotting, and in situ hybridization. So every technique has its own principle. Okay, so ultimately to find out the gene expression, we are going to use all these options. So out of this, two primary methods are the microarray and RNA-seq. So the microarray is this is generally used to use pre-designed probes to measure the expression levels of thousands of genes in thousands of genes simultaneously. Okay, so for example, for RNA-seq, RNA-seq is a next generation sequencing method that provide the complete picture of complete unbiased picture of gene expression by direct sequencing of the RNA molecule. So ultimately, the both generated data set is significantly helpful for us to finding or for detection of differentially expressed genes. So here, for example, if you see the output of the raw or output or raw data from the gene expression experiment, it is actually the starting point for all the downstream analysis. So these data files are generated directly by instrument during the hybridization or sequencing process, are stored the intensity values or count representing the gene expression levels. Each platform, you can see here various platform I mentioned here, each platform such as Affymetric, Agilent, Illumina produces data in a specific format. Like if you see this Affymetric, they are providing the CL format file, and other platform you can see there's one, there are different formats they are going to provide for you. Okay, so for this one, which definitely contain the unique structure suitable for their respective analysis pipeline. These format are required specialized tools. Okay, it's every format requires a specialized tool or software to extract the meaningful information all to perform the final product is a differential express gene. For example, once more time I'm give an example for you. For Affymetrics, we will have the CL format file typically processed. Generally we will process by using the R packages like Affymetry, Affy package, or Lima package, or Oligo package. So these are the general packages generally we use in R to get the differential expression. For example, in Agilent condition, you will get a text format file where you have to open in a spreadsheet software. So that handling these files is critical because raw data is often the noise and requires multiple processing steps such as background correction, quantity control, normalization to ensure this that whatever the interpretation you are going to give you, it should be accurate. Okay, so if you see the overall workflow of the gene expression, how exactly we can do it from the scratch, from initial sampling to end of the biological interpretation. So the gene expression analysis workflow involves the series of well-defined steps. Okay, so in beginning, so we have to extract the feature from depends upon the type of instrument we are using. So where, for example, if you take the CL format file or IDAT format file, where the raw data intensity value from the files like a CL and IDOT are converted into usable data sets. Okay, so usable data set. So the next step is we have to check the QC, the quality control, to evaluate the reliability of the data, to identify the potential outlier or technical errors in the in the experiment. So next step is we have to normalize this data to adjust the bias or batch effect, ensure that the data from the different samples are comparable, and the heart of the workflow is the next step is the differential expression analysis, where we have to use a different type of statistical method to identify the gene that are significantly upregulated or downregulated in between the groups. The finally, we have to subject it for the biological interpretation, which include the gene clustering, Go enrichment analysis, pathway analysis. It will give a clear understanding for us to identify the functional roles of these differentially expressed genes. Okay, so overall, this workflow provide us a structural approach to analyze the gene expression data, offering insight into underlying biological mechanism of disease pathways. Okay, for the gene expression data submission and retrieval, there are many databases are available. So the major one are the GEO and ArrayExpress. Okay, so these are the two major repositories are there. These repositories consist of a variety of gene expression dataset or transcription datasets information from different species, and they are having diverse experimental condition also. Okay, so let us explore these two. GEO, generally GEO is hosted by the NCBI, and it provides the vast repository for the transcription data like a gene expression data. Okay, let us see how we can access this data. Okay, let's to go to the Google and type here NCBI GEO. Okay, so click enter. So you will find the first page of NCBI GEO. Okay, so click here, and here it consists of the search box, the major search box, where you can see you can search by using the different type of keywords. Okay, so for timing, I'm just typing the one small keyword like a cancer. So if you type the cancer, so here you will automatically redirect into two different things for you. One is geo dataset and geo profile. We are looking for the dataset main. Here you just need to click this geo datasets. So now you can see here there are so many results we got it. So many results with the name of the cancer. So you always have a, if you want to further filter down, okay, further filter down based on the organism name, based upon the what type of tissue you are using, what type of experimental condition you are using. Okay, you can use a different platforms or different filter conditions to narrow down the results. Okay, or if you already know that know the dataset ID, for example, if you go back here, I already know one dataset ID. Okay, for example, GSC 115810. Okay, so I'm using this dataset. If you click this one, so it will take us to the database page, datasets page. So where you will find the detailed information such as experimental performance, the platform use, sample number, okay, and sample number, whose authors are there, which journal they published. Okay, you will find the citation also here, and you will find the overall design of the workflow. Like here we have nearly 27 samples are there, 3 are control sample, and different phases in the cancer, nearly 7, 11, and 6. Okay, 23 are 23 samples are there, or 24 samples are there, which is for the cancer. Okay, so and also you will see who exactly, from where exactly this data set has been collected. So if you go to the bottom, you will find the option called as a download data. Okay, you can always have it download this data in the raw format. Okay, you can download the data in raw format. Once you download the raw data format, you can use this one to run in your local machine. So there is one more option is available. If you go to the top of the page, here the analysis option is available, where if you want to directly analyze this data, you can click here. Simply it will redirect to the GO2R, where you can perform the gene expression analysis directly from here. So second database we are going to talk, that is ArrayExpress. Okay, so we just need to type the same thing here, ArrayExpress. So it will redirect to the first page. It's RxPress one. So RxPress is majorly hosted by this EMBL EBI. It's one more very good repository for the transcriptome or gene expression data. So here uh, you can also have this search bar. You just need to type the keyword here also, the same thing. Like so I'm going to type the same keyword like cancer. So here, once you click on the cancer, you will find the different data sets are there. So I'm going to select the first one. So that is a epigenome analysis of normal and cancer tissues in a prostate cancer. Okay. So if you click here, so here you will find a detailed description about the dataset, including the organism name, platform, sample details, experimental condition. Okay, all those information you can easily find out here. A unique feature in the ArrayExpress is you will have this detailed sample information linked to the data. If you click this one, where you can find a detailed data points like a disease state, tissue types, and labeling method. So all those things you can easily find out by using this option. So the only disadvantage with the ArrayExpress is you cannot directly perform any analysis here. You have to download the raw data directly from here, and once you download the raw data, you can run this raw data in your local machine. Okay. So let's go back to the original analysis, where we are going to work on the GO2R. So in web-based platform to perform the differential express genes. Okay, to perform the to identification of a differential express gene from the already available data sets. Okay, so here click on GO2R. So click the first link. Okay, so here you have to type again the same dataset ID. Okay, so GSC 115810. Okay, so I'm going to use the same of this one. So if we click on the set, so it will load the data set and show the study design and the sample type and all the other metadata for you. Okay, so to perform the DEG analysis, you need to define the specific groups. So you have to review this metadata to identify the what are the controls are there, what are the cases are there. So you can see here clearly, here we have the three controls are there and other all are the affected groups. So in defined group, you have to create two groups, as I said, to control and affected group. So click here the control group and affected group. Okay, so once I click the once I assign the control and affected group, so then we have to assign the sample for each group by selecting them and click the appropriate group names. Okay, so here what we have to do is we have to select the control group first. Okay, for example, these three controls I'm selecting. Now I am assigning to the control. Okay, so once you click that one, it will show you the how many number of samples you are having in the control. Similarly, for the affected group, I just hold the shift button or selecting all the samples. Then I click on the affected one. Now it is showing the 24 samples here and 23, so 3 samples of the control. Now uh, now you can have now you just need to go for analyze this one. Okay, so here you can click on analysis directly, but you have options here. Few options are there, like a statistical options are available, where you can change the or adjust the p values, or if you want to show the plot or display or images with the particular threshold, you can always have a option to change it. Okay, I'm just keeping everything as a default and click on analyze. It doesn't take much time for you. It will take hardly 1 to 2 minutes to display the results. So once you display the result, it will show you the clear information for you, the differentially expressed gene. So you can see here initially we have to see this result table which displays DEG with the column like here ID. ID is nothing but the probe ID. Okay. So the what exactly the probe ID is in gene expression experiment, a probe ID is a unique identification number, okay, for the specific probe on the microarray chip. For each probe bind to the specific gene or transcript, it measures its expression level based on the intensity of the signal generated. Okay, the probe ID it need to map to the specific gene name or the symbol using the annotation file. Okay, so it automatically do this annotation in a background. Okay, some genes you will find some genes may have the multiple probes. Some genes may consist of the multiple probe. So you have to carefully consolidate the analysis. Okay, you have to check the analysis accurately based upon the if the multiple probes are there, you have to take the average of the log fc value. Okay, so and also you will find the other values like a log fc, and you will find the statistical values like adjusted p values, a gene symbol, and also gene titles also you will find it. Okay, so and also along with this one you will also find different visualization patterns also, like you can see the volcano plots are there, and you will also find this mean difference plot. You will find if you are using the multiple data set, this Venn diagram is also useful for you, and you will find the intensity plots also is there. For normalization results, you also have this box plot. You have p-value plot and other statistical plots also is available for you. Hit in the display, it will show you only certain significant genes only. It is not going to reveal you all the significant genes. For this, you have to download the full table. Okay, so you have option to download the full table. Once you click on the full table, this will allow you to download the full table. Okay, so I'm just downloading the full table here in TSV format. So from here you can use a different filter option, okay, to sort down the genes based upon their fold chain. Okay, so we need to consider two parameters, especially here, the log fc and the p value. So these two are the major parameter for us. Okay, so here first of all we have to check the log fc. You can sort them with the different sort. You can sort them either by highest to lowest or lowest to highest. Okay, for example, in this data set, uh, I'm going to use the lowest to smallest to largest. If I use the smallest to largest, you will find the fold change of minus 2.93 for the gene TFF3. Okay, so vice versa, I will do some largest to smallest. So in a largest to smallest, you have find the log FC of 3.4 for the SFRP4 gene. Okay, that is secreted for fertilized protein 4. So So these upregulation and downregulation are reflects in the positive and negative log FC values. Okay. So understanding the behavior of these DEGs are crucial uh, crucial because these changes in expression level help us to identify the significantly differential expressed gene in this data set. Okay, so let us discuss bit theory behind the log FC, why it is very important in the gene expression analysis. So the log FC value actually measures how much gene expression changes in between the two groups, such as cases and controls. Okay, and also it will calculate it. It is going to calculate it. It is calculated using the formula. Okay, log FC is equal to log square. Okay, is average expression of cases, or it means average expression in the affected groups divided by average expression of the controls. If the log FC is plus 1, it means the gene is expressed 2 times higher in the cases compared to the control. Vice versa, If log FC is -1, it means the gene expression is 2 times lower in the cases compared to the control. Okay, these values gives a clear understanding whether the gene is upregulated or downregulated. But similarly for the p-value indicates the statistical significance of the observed different genes in the gene expression. It will tell us whether the changes in the expression are likely due to chance or genuinely it is significant. Okay, always the smaller p-value indicates the changes are statistically significant. Always we consider in a gene expression analysis less than 0.05. In next video, we'll use the same spreadsheet to filter down significantly expressed genes and focus on these genes to explore their biological role. So this will include the performing the Go annotation and pathway analysis using the enricher tools and also the cytoscape or Gluco tools. Okay, so thank you so much for joining. Make sure you subscribe and hit the like button. This is always encourage us to create more or more content like this. This is Dr. Babachan Badanganapalli and you're watching Bioinformatics with Baby.